So no introduction to the field of microbiology would be complete without talking about some of the tools of the laboratory, which is covered in Chapter 2. So we're going to talk about some of the tools that you'll be working with in lab this semester to help you better understand microbiology. So in your lab that accompanies this class this semester, as well as microbiology labs all around the world, we use 5 different techniques to culture and grow and study microorganisms. And so those 5 different techniques are the 5 I's: inoculation, incubation, isolation, inspection, and identification. And of course, we'll talk about each one of these a little more in detail, but a great way to visualize these 5 I's together is in figure 2.1 in your textbook, and it's a nice visualization of what each one of those steps is. So let's start with the first I. Step 1 is inoculation. And so in order to grow organisms, we need to first start with some type of sample of the microbe. Okay, remember, biogenesis from our previous lecture means that living organisms come from other living organisms. So the only way we're going to get more microbes is if we start with some microbes to begin with. And so the sample that we start with of the microbe is called the inoculum, and it is a tiny sample of the microorganism. These guys are tiny, so you just need a little bit to get going. And the inoculum is introduced into the microbial food, which is called media. So I would definitely circle inoculum on this slide. I would circle media. These are important words you need to know. So the microbial food is called the media, and we put the inoculum in the media, and that is called inoculation, the process of introducing the inoculum into the media. And so when performing this process, it's really, really important that the instrument you use to inoculate the media itself, other materials you're using, it's important that those things are sterile in order to prevent contamination, meaning growing other organisms you don't want to grow. You only want to grow what you have in the inoculum. And so this is really the most important first step. You're just getting that microbial culture started by taking a sample of the original culture and introducing it into media, and that process is called inoculation. The second I is incubation. So step 2 is incubation. So once we've performed our inoculation in step 1, we need to put that media that has the microbe inside it into what we call an incubator and incubate that culture. So incubation is the process of taking the culture, which is the medium plus the microbe, and putting it in a temperature controlled chamber known as an incubator. And this is a really important step because most microbes grow at temperatures slightly higher than room temp. So the incubator is going to make them nice and happy and get them growing really well. Now this process is typically a 24 to 48 hour process, but it can take much longer with slow growing organisms. So when those first 2 I's combine, we get microbial growth. So inoculation plus incubation is going to give us growth. And this microbial growth can manifest in a variety of ways. It really just depends on the type of media that we're using. Remember, the media is the food, or the type of organisms that are involved. So in a liquid media, which sounds just like what it is a liquefied version of the media. It's, a liquid. It's not solid, it's not gas, it's a liquid. And so in a liquid media, microbial growth is going to look like cloudiness, like sedimentation, there's chunks floating around, or it can turn the media colors. On a solid media, though, microbial growth appears as colonies, and you can see that on the slide. A colony is what the blue arrow is pointing to in the left-hand corner of the slide. That is what a colony is. It's microbial growth on a solid media. We don't see colonies in a liquid media, okay? So cultures can be found in 3 main types of conditions. It can be as what we know as a pure culture, which only contains a single microbial species. And that's what we see on the photo above that box. We see on the left-hand side those 3 tubes. Those have pure cultures with only 1 type of microbe in each. We can have a mixed culture, which contains multiple organisms originating from the same source. For example, say we had to swab a wound or swab a mouth. That's going to be an area of the body that might have more than 1 type of organism growing there, and so we could potentially get a mixed culture, something that's going to have more than 1 type of organism growing there. We can also have what we call a contaminated culture, and that's a culture that was once pure or mixed, but now has unwanted contaminating microbes introduced into it, either during inoculation or during incubation. And so it could be a pure culture or a mixed culture that now has other organisms that didn't originate with that original inoculum. And you'll have a chance to grow your own pure cultures this semester during lab time. Okay, let's talk a little more about microbial food, what we call media. So just like human food, there's a lot of different choices out there. There's a lot of different choices of microbial food out there as well. And so Table 2.1 nicely summarizes the 3 main categories of media classification. And we can break the different types of media up out there into the physical state that it's in, the chemical composition that it has, or the functional type. So I just mentioned a couple of physical states of media on the previous slide. I was talking about how microbial growth manifests as cloudiness in a liquid media and how it looks like colonies on a solid media. So let's talk a little more about those different physical states. So the physical state has a lot to do with the amount of agar that is added to the media. And so agar is a solidifying agent. Agar is a very important term, by the way, so you need to make sure you know that. And so what it is, how much agar is in it determines how solid the media is. And check out figure 2.3 if you really want to know what these physical states look like. And so if there's no agar in it, we're going to call that media a liquid media. If it has a small percentage of agar in it, we're going to call it a semi-solid. It's going to be like a gel. And then a solid is going to have a pretty good amount of agar in it. And so we can have solids that can be converted to liquids and solids that cannot. In terms of chemical composition, we have 2 main groups. We have a chemically defined group, which has the exact known measures of chemicals in the media, and then we have complex media that is not chemically defined. So complex media contains at least one ingredient that does not have an exact chemical formula. For example, something like blood serum. That's going to vary from batch to batch, so it's not going to be an exact chemical formula. So a complex media is going to contain at least one ingredient that we can't say we know exactly every single item that's in it. Now the functional type is going to be basically based on what do we want to use this media for. Depending on what we're trying to do by growing the organism will help us determine what kind of functional type we need. Different types of media are used for different purposes to help narrow down the identity of a microbe. So for example, a general purpose media, that's going to grow pretty much anything. It's not going to differentiate between one type of organism or another. It's not going to keep certain organisms from growing. It's just going to be a pretty much catch-all that will help anything grow. But then we have other things like enriched and selective and differential media, and those will grow only certain types of organisms. And we'll talk more about that during lab time, but it's important that you understand that we have different types of media. They're used for different applications. So back to our 5 I's. Remember, we had an inoculation and incubation as our first 2. Step 3 is going to be isolation. In order to accurately identify a microbe, we need to have it isolated from other types of organisms. That just makes sense, right? It's hard for us to be able to identify something unless we kind of have it by itself and we know, hey, this is just one microbe alone. It's not several microbes together. And isolation is best accomplished by trying to grow isolated colonies of bacteria. So remember, we mentioned the word colony before. It's what we see on a solid media on an agar. And a colony is just a discrete mound of cells that is formed from one individual bacterial cell dividing over and over again and growing into this nice little lump that we see on solid media. So isolation is best done through growing isolated colonies. And we use different types of isolation techniques. We use the streak plate, we use the loop dilution or the pour plate, and we use the spread plate. And again, we'll talk more about those in lab this semester, but it's important that you understand microbiologists have a few tools in their tool belt. They don't just have to do things one way. There are several different ways that we can isolate microbes. So the last 2 I's are steps 4 and 5, inspection and identification. So once we've inoculated, incubated and isolated. Now we want to look at those cultures, those colonies. We want to look at them macroscopically. We can learn a lot from what a colony looks like, its morphology, meaning what color is it, what texture is it, what do the margins look like, and you'll do that in lab this semester. We also want to look at those specimens microscopically. So we will prepare a culture, put it on a slide, and look at it. What does the cell look like? What shape is it? What size is it? Does it move? That type of thing. So we're looking at inspection. And then we also want to identify. So we'll run a battery of tests to try to identify the organism. We'll do things like cell staining, biochemical tests, immunological tests, and DNA analysis. And the results from these tests will try to tell us what microbe we are working with. So we'll do an analysis of those results and try to pinpoint the identity. And of course, this usually takes doing a few types of tests. We don't just, do one type of test and we know for sure. So we have to do some cell staining, growing up some different types of media, differential media, selective media. So identification can be a little bit more time consuming than the other steps. So an extremely important tool of a microbiologist is the microscope. In fact, you can think of it as the microbiologist's best friend. So of course, we've been talking about microbes, that the fact that they're tiny, they're prokaryotes, they're really small, and so we're going to need a microscope to see them. Now, primarily we use a light microscope for most microbiological applications, but there are other microscopes that exist out there that we can use for certain types of specimens. Microscopes like the electron microscope, or a confocal, or fluorescent, or phase contrast microscope. Those tend to be more expensive and used for more specialized applications, but we can use those as well. But in general, we're going to use light microscopes, and that's what we'll use in lab this semester. Now, the main goal of microscopy is to magnify or enlarge while keeping good resolution. And resolution is the ability to distinguish between 2 points, and that determines how clearly we can see something. So we want to be able to see it clearly, but we also want to be able to see it enlarged. And that's what a microscope does for us. So we're going to take a moment and do a clinical focus to kind of apply this information we've been talking about. And we're going to apply it in terms of collecting patient samples. So the process of collecting patient samples is a critical step in the process of growing and identifying microbes, which is what we've been talking about today. So if we have an improperly collected sample, we can get contamination of the specimen, and that can lead to problems later on. And so this contamination generally comes from the normal microbiota of the patient or from contaminants from the collector. So that would be something you'd want to keep in mind as a collector. And then contamination can lead to failure to identify the pathogen accurately, right? If you've got other things in there, you can't be sure if it came as a contaminant or if it came from the specimen itself. And so that can also lead to problems with treatment for the patient. And then when collecting patient samples, it's really important that you follow all the standard protocols to maintain this integrity of collecting patient samples. So you need to clean a wound prior to collection, or ensure that patients follow clean catch urine sample procedures if that's what the procedure calls for. So following those protocols will definitely help you make sure that you collect a patient sample correctly and that it passes along the 5 I's in the proper manner.